morning, everyone. Welcome to the second KDOC Science Online Conference on extracellular vesicles and extracellular vesicle-based therapeutics. I am In-Sun Kim from Korea Institute of Science and Technology. I'm hosting this conference, organized and sponsored by KDOC and ShiftBio. Today we have two sessions. One is in the morning. The second one is in the evening. We have two speakers in the morning session and two speakers in the evening session. I will give you a brief introductory presentation. So why we need to pay attention to extracellular vesicles and exosomes. This is my disclosure. Let me simply classify disease into two groups. One is a simple disease. The other one is complex disease. Simple disease is just like amputation, dehydration, infection, etc. As we live longer, we may have more complex and chronic disease than before. The simple disease, here I give you one example, hypercholesterolemia. Of course, this is not as simple as amputation or dehydration, but I think it's relatively simple compared to neoplasm or other complex disease. To cure the hypercholesterolemia, we need to target one key enzyme, which is very critical in the biosynthesis of cholesterol. That's why we have statin. Whereas the complex disease is very much complicated with so many different molecular pathways, as well as many different types of cells. So we definitely need to develop multi-targeting and multifunctional new therapeutic modalities. So today I brought issues that require new therapeutic modalities for the treatment of complex disease. The first one is undruggable targets. The most undruggable targets are intracellular or membranous. As you can see in the diagram on the left side, more than 50% are intracellular and more than 30% are membranous undruggable targets. So we do not have a low-hanging targets left so far. We have very hard targets. These undruggable hard targets are typically intracellular and require large key molecules, such as a protein, rather than a small chemicals. But these large keys are usually not permeable. So the question here is how to deliver them into cells. The second issue is its complexity of disease, as I mentioned before. The complexity of this chronic disease is going to handle. It is very hard to handle. The chronic disease, because the chronic diseases are very complex and heterogeneous and polygenic, but their diagnosis are often based on similar observed symptomology. Even these symptoms may result from multiple different pathways. So the question is how to target multiple pathways at the same time or sequentially. On top of that, we have another big problem. Chronic diseases are progressively making evolution, often in unpredictable ways. So here's the second question, how to come up with this disease evolution? And so the issues are cells as a target. So many different types of cells are involved in complex disease, like here is ARDS. Type 1 and 2 alveolar cells involved. alveolar macrophages is involved, and fibroblasts, neutrophil, platelet, and RBCs, etc. So many immune cells are also involved in this process. So the question is how to orchestrate all these different types of cells. Here I give you more questions in detail. How to remove diseased cells, how to reprogram cells to repair themselves, how to regenerate parenchymal cells, how to reactivate exhausted cells, and how to repress overactivated cells. Of course, we want to hear the best sound of world's best orchestra, but the disease is different. We just want to survive disease, and then we want to be okay as it was. So it is a very tough task to overcome this very complicated disease. But fortunately, we have a wonderful self-healing system within us. So what the Hippocrates said a long time ago, we have natural forces within us, and this is true healers of disease. So now we just need to help our natural forces within us to do their job best. So the last issues that I brought today is intracellular regulators as therapeutics. Most drugs are inhibitors or antagonists. Here's an example. We have many different types of antiplatelet drugs, but all these antiplatelet drugs are inhibitors of one of these four pathways, ADP, or COX, or phosphodiesterase, or GP2B and 3A. So why don't you have a kind of activator instead of inhibitor? But the problem is most activated could act in the cytoplasm or in the nucleus. So most key factors are intracellular. But we really need to have these intracellular key regulators as therapeutics. So the problem is how to deliver, how to get them into target cells. So to have this multifunctional activity, we definitely need to deliver key factors into target cells. For example, here, HIF1 alpha, which is a very critical transcription factor in the regulation of metabolism, perfusion, and growth control, etc. So we can, if we can deliver these key factors into target cells, that would be wonderful. But it is extremely difficult to deliver this key factor. So how to answer all these questions? That's why we have a conference on exosome today. Every single cell can produce membrane structure, extracellular vesicles, and exosomes. As you can see here, the exosome can carry many different types of membrane proteins, as well as many cargos inside, such as proteins and nucleic acids like DNA, messenger RNA, and microRNAs. So these exosomes are perfect nanovesicles for membrane protein delivery, not only membrane protein delivery, but also many other active ingredients. So I will briefly introduce the beauties of exosomes. First, the exosomes are very biocompatible. As I said before, every single cell, not only human, but also from bacteria, every single cell is supposed to secrete extracellular vesicles. So it means it's very biocompatible. And second one is exosomes immunologically tolerable. if you think about that, how many exosomes are actually present in the blood. It's about 10 billion exosomes are present in just 1 ml of blood. But it doesn't cause any problem when you have a blood transfusion. So it is immunologically very tolerable. And it is natural membrane structure. So basically it can carry many different types of membrane proteins, particularly multi-membrane, transmembrane domain membrane types of proteins. And we can also load many different types of active ingredients by naturally or genetically or physicochemically. And we can also modify surface of exosome. We can make it targetable, or we can make it fusogenic, just like a virus, or we can make it to have more long half-life and better biodistribution, and then it also penetrate a blood-brain barrier. So we can combine the above functions into one nanoparticle, one exosome. So it could be multifunctional.